We're talking about the molecular machinery of life,
Mhm.
specifically focusing on muscle contraction and the cytoskeleton.
It's the internal combustion engine of the human experience, really. And the hook, I think, the thing that should really grab you, is how we normally see muscle. We see it as, well, as meat.
Yeah, you go to the butcher, you buy a steak. That's muscle.
Exactly. But if you were to put that steak under a powerful enough microscope, you'd see that meat is this unbelievably precise, synchronized array of billions and billions of tiny molecular motors.
All called myosin, all pulling perfectly in unison.
It's a miracle of coordination.
And it's not just about the big muscles we think of, right? Like biceps or quads, the skeletal muscles. The source material really hammers home that this machinery, this toolkit, it's universal.
Absolutely. It's inside the heart, which is maybe obvious. It's lining your gut. But it's also inside every single one of your cells, forming this internal scaffolding.
The cytoskeleton.
Cytoskeleton. It's the only thing that keeps us from collapsing into a puddle of protoplasm.
So without this internal framework of actin and tubulin that we're going to get into, you wouldn't be a complex organism. You'd just be...
You'd be an amoeba at best. And what's truly terrifying, which the text briefly mentions, is that this machinery is so fundamental to how we work that other organisms have figured out how to weaponize it against us.
Oh, you're talking about that note on the bacteria, Listeria and, was it Shigella?
Listeria and Shigella, yes. And these are not passive germs that just float around making you sick. Listeria monocytogenes, for example, literally breaks into one of your cells and hijacks your cytoskeleton.
What do you mean hijacks?
It grabs your own actin molecules, the exact same protein you use to flex your arm, and it starts building a rocket tail behind itself.
No way.
It polymerizes the actin into a little jet engine to propel itself through your cell's cytoplasm at high speed so it can punch through into the next cell and infect it. It uses your engine to invade you.
That is the literal definition of a hostile takeover.
Ah!
Okay, so our mission for this deep dive is to decode this engine. We need to get into the physics of the movement. We're going to break down something called the power stroke, which it sounds like an Olympic rowing term, but it's actually quantum level geometry.
It really is.
We have to look at calcium, which seems to be the master on-off switch for everything. And of course, we have to talk about what happens when the machine breaks down.
And the clinical correlates here they're serious. We're going to be talking about muscular dystrophy, which is basically a chassis failure.
Okay.
We'll get into malignant hyperthermia, which is a catastrophic engine overheating event. And even something like progeria, which is rapid aging, turns out to be a problem with the structural lining of the cell's nucleus itself.
It's a huge amount to cover. So let's set the stage first. The text lays out a really clear hierarchy of muscle types. We have skeletal, cardiac, and smooth. I think we need to get the basic specs on the table before we start tearing them apart. So start us off with skeletal muscle.
Okay, skeletal muscle. This is your classic marquee muscle. It's the stuff that you use to move your skeleton around, hence the name. The key features are, it's voluntary, meaning you have conscious control over it. If you decide to high-five someone, your brain sends a signal and your skeletal muscle executes the command.
And what does it look like structurally?
Structurally, and this is critical, it is striated. If you look at it under a microscope, it has stripes, these alternating light and dark bands, and those stripes are the secret to how it works, which we'll get to.
Okay, voluntary and striated. Next up, cardiac muscle, the heart.
Also striated. So under a microscope, it looks very, very similar to skeletal muscle. It has those same stripes, but functionally, it's a completely different beast.
Oh, so?
Well, for one, it's involuntary. You can't just decide to stop your heart, and it has to pump rhythmically 24/7 without any rest for your entire life. It has its own pacemaker.
No pressure at all. And then finally, we have the outlier of the group, smooth muscle.
The quiet workhorse of the body. Smooth muscle is what lines all your hollow organs, your blood vessels, your stomach, your intestines, bladder, uterus.
And the key difference is in the name.
Exactly. It is not striated. That's why it's called smooth. It looks much more uniform under the microscope. And, like cardiac muscle, it's completely involuntary. You don't consciously tell your stomach to digest your lunch. It just does it.
And the actual mechanics of how smooth muscle works are totally different, aren't they? It's built for a different kind of job.
A completely different job. Skeletal and cardiac are built for speed and power, big, rapid contractions. Smooth muscle is built for efficiency and for holding tension over long periods.
Like what?
Like keeping your blood vessels slightly constricted to maintain your blood pressure all day long. It's the difference between, say, a piston firing in a car engine versus a clamp holding two pieces of wood together. Smooth muscle can enter a latch state and hold a contraction for hours without burning through all your energy.
A clamp versus piston. That's a great way to think about it.
Okay.
Specs are established. I want to dig into skeletal muscle first because the text really uses it as the baseline model for understanding all the mechanics.
It's the best place to start.
It describes the architecture of the muscle cell in a kind of Russian nesting doll situation. Can you help us visualize that? If I were to slice open my bicep, what am I actually looking at from big to small?
Okay, so let's zoom all the way out. You have the muscle belly itself, the whole bicep. That's wrapped in a tough sheath of connective tissue. Now, if you slice that open, you'll see it's not one solid block of meat. It's made of bundles.
Okay.
These bundles are called fasciculi. Think of them like the thick, wound-up cables you'd see in a suspension bridge.
And inside each of those cables, the fasciculi.
Inside those, you have the individual muscle fibers. Now, this is a really crucial conceptual leap for the listener. In normal biology, when you hear fiber, you think of a thread, something non-living.
Right, like a piece of string.
But here, a muscle fiber is a single living cell.
But it's not a normal cell, is it? It's huge.
It's a giant, a monster. A single muscle fiber cell can be centimeters long, sometimes running the entire length of the muscle. And because it's so enormous, one nucleus, the little brain of the cell, isn't enough to manage it.
So it has more than one.
It has hundreds, sometimes thousands. Muscle fibers are multinucleated. During your development, hundreds of little baby cells called myoblasts all fuse together to create this one giant megacell.
So it's a syncytium. It's like a cellular collective.
That's the perfect term for it. It's a syncytium. And inside this megacell, floating in the cell fluid, which in muscle we call the sarcoplasm, you have thousands upon thousands of even smaller threads.
Okay, so going deeper.
These are the myofibrils. And these are the actual contractile elements. This is where the work gets done.
And we can keep zooming in, right? If we take an electron microscope and point it at one single myofibril.
Then finally, you see the engine block. You see the sarcomere. The text defines the sarcomere as the fundamental functional unit of muscle.
The basic repeating unit.
The basic repeating unit. It's tiny, only about 2 to 2.3 microns long. But this little segment is where all the magic happens. If you can understand the sarcomere, you understand all of human movement.
All right, let's map out the sarcomere. This is where we get that striated or striped look we talked about, correct? It looks like a barcode.
It looks exactly like a barcode. And just like a barcode, the pattern of light and bark and the spacing tells you everything you need to know.
So what are the parts?
The sarcomere is defined as the space between two walls. These are called the Z-lines or Z-discs.
Why Z? Is there a reason for that?
It comes from the German Zwischenscheibe, which just means disc in between. So the Z-line is the anchor point. It's a protein scaffold at each end.
So we have two walls, the Z-lines, facing each other.
Exactly. And attached to these Z-lines are what we call the thin filaments. They stick out from the Z-lines and point towards the center of the sarcomere.
Okay, so imagine two walls with ropes hanging off them, pointing towards the middle but not quite touching.
A perfect analogy. And floating in the very center of the sarcomere, suspended between the ends of those thin filament ropes, are the thick filaments.
And the way these thick and thin filaments overlap, or don't, is what creates the bands we see under the microscope.
Precisely. The darkest stripe in the barcode is called the A band. The A stands for anisotropic. This band corresponds to the full length of the thick filaments.
Okay, so wherever there are thick filaments, that's the A band. It's dark because there's a lot of protein packed in there.
A lot of dense protein, yes.
Yeah.
Now, the lightest stripe is the I band. I for isotropic. That's the region near the Z-line where you only have thin filaments with no overlap from the thick ones.
So it's less dense and more light gets through. Makes sense.
Mm-hmm. And the text also mentions one more, a little subtler, the H band.
Right in the dead center of the A band.
Exactly. The H band is that little gap in the very middle of the sarcomere in the center of the A band where the thin filaments haven't reached yet. It's H for heller, which is German for brighter. It's just a little less dense than the rest of the A band.
Okay, we have the geography down. Z lines, A band, I band, H band. Now, the movement. This is the whole point of the sliding filament model.
Yes.
For a long time, scientists thought muscles contracted by, bunching up or coiling like a spring or an accordion.
Which is a totally intuitive idea. If you see something get shorter, you naturally assume the material itself is shrinking or compressing.
But the sliding filament model proved That was completely wrong. When a muscle contracts, the filaments themselves, the thick and the thin, they do not change length at all.
That is such a wild, counterintuitive concept to wrap your head around. The entire muscle gets shorter, sometimes by a lot, but the individual parts that make it up stay the exact same size.
So how does it work?
Think of it like a telescope extending and retracting. Or an even better analogy is to interlock your fingers. Hold your hands apart, palms facing you, then slide the fingers of your right hand between the fingers of your left.
Okay, I'm doing now.
The total hand span from your left pinky to your right pinky just got a lot shorter.
Yeah, by more than half.
But did your fingers shrink?
No, of course not. They just overlapped more.
That's it. That's muscle contraction.
Okay.
The thin and thick filaments slide past each other, increasing their overlap.
And we can actually see the evidence for this under the microscope, because the bands change when the muscle contracts.
This is the visual proof. This is the smoking gun. When the muscle contracts, you can watch the two Z lines get pulled closer and closer together.
Okay, the whole sarcomere shortens.
The whole sarcomere shortens. The I band, that light part with only thin filaments, gets much narrower. The H band in the middle, that gap, disappears entirely because the thin filaments slide all the way to the center.
And this is the key.
But the A band, that dark stripe that represents the length of the thick filament, it stays exactly the same length. It doesn't change one bit.
Because the thick filament isn't squishing or shrinking. It's just the thing doing the pulling, and the thin filaments are sliding deeper into its territory.
Precisely. The fact that the A band length was constant was the undeniable evidence that killed the old shrinking spring theories and proved the sliding model.
So we have the chassis and the telescoping mechanism. But a telescope doesn't close on its own. We need a motor. We need to actually meet the players that are driving this slide. We do. We have the thick filament and the thin filament. Let's start with the heavy lifter, the motor itself. Myosin II.
Myosin is a molecular beast. It's a truly massive protein complex. The text calls it a hexamer, which means it's built from 6 separate protein chains.
6 chains.
2 big heavy chains and 4 smaller light chains.
But for understanding the movement, we mostly just care about the head part of it, right?
Yes, that's where all the action is. Imagine a 2-headed golf club, or maybe a 2-headed snake. The long tails of the 2 heavy chains wrap around each other to form the thick central rod of the thick filament.
Okay.
But at one end, the chains separate and fold up into these 2 globular heads that stick out. That head is the motor, and it has 2 absolutely critical features.
What are they?
It has an actin binding site. Think of that as its mouth, what it uses to bite onto the thin filament. And it has an ATP binding site. That's its fuel tank.
And the text had this really interesting little historical aside about how scientists figured this out. They used meat tenderizer, basically. They used digestion enzymes.
This is just classic old-school biochemistry at its finest. It's so clever. They took the big myosin molecule and they digested it with enzymes like trypsin and papain.
And what did they find?
They found they could chop the molecule into specific pieces. They could chop the long tail off, and they called that light meramyosin, or LMM. The piece left over, which had the 2 heads and a bit of the neck, they called heavy meramyosin, or HMM.
So they separated the head from the tail.
And then they went one step further. They used another enzyme to chop the heavy meramyosin again, right at the neck, to isolate just one single head. They called it the S1 fragment.
And that S1 fragment was the key.
It was the key, because they could put just that single protein fragment in a dish with actin filaments and ATP, and it still worked. It would still bind, pull, and release. It proved definitively that the entire motor function is contained within that one single head.
Wow. It's like taking the engine block out of a Ferrari, putting it on the garage floor, connecting the gas line, and watching it vibrate all by itself.
That's a perfect image for it. The power is all in the head.
So that's the motor. Now we need the track it runs on, the thin filament. This is primarily actin.
Actin is a really interesting protein. It exists in 2 forms. It starts as individual little globular balls of protein, which we call G-actin.
Okay.
But in the cell, under the right conditions, these little G-actin monomers polymerize. They snap together like Lego bricks to form a long, twisted, double helical rope. And we call that F-actin for fibrous actin.
And that F-actin rope is the track that the myosin motor runs along.
It is the track, but it can't just be a naked rope, because if it were, the myosin heads would just be grabbing onto it constantly.
All the time.
You'd be in a permanent state of full-body cramp. There would be no relaxation. You have to have control system, a safety switch.
And that safety switch is the first of our regulatory proteins, tropomyosin.
Yes. Tropomyosin is another long, fibrous, rope-like protein. Its job is incredibly simple, but absolutely vital. It winds around the F-actin rope, and it physically covers up the active sites where myosin wants to bind.
So it's like putting electrical tape over all the keyholes.
Exactly. It's the gatekeeper. It blocks myosin from binding to actin.
And then there must be something that controls the gatekeeper, the lock on the gate. And that's the troponin complex.
Right. Troponin is this little cluster of 3 smaller proteins that sits at regular intervals along the tropomyosin strand.
3 parts.
3 subunits. You have troponin T, where the T stands for tropomyosin. Its job is to tie the whole troponin complex to the tropomyosin rope.
Okay, the anchor.
Then you have troponin I, where I stands for inhibitory. This subunit helps hold the tropomyosin in that blocking position over the active sites. It's the inhibitory part of the lock. And then you have the most important one, troponin C.
And I'm guessing the C stands for?
Calcium. Troponin C is the calcium sensor. It's structurally very similar to another important calcium-binding protein called calmodulin. Troponin C has these 4 little pockets that are perfectly shaped to catch and bind calcium ions.
Okay, so let's put the whole control system together. Let's say the signal to contract arrives. What happens?
The signal causes a flood of calcium into the cell. Those calcium ions find their way to the troponin C subunits and bind to them.
Right.
The binding of calcium causes the troponin C protein to undergo a major shape change, a conformational change. It twists.
And because it's tied to the other parts.
Because it's tied to troponin T and troponin I, which are tied to the tropomyosin rope, that twisting motion physically yanks the entire tropomyosin strand sideways.
It pulls the electrical tape off the keyholes.
Exactly. The binding sites on actin are suddenly exposed. The myosin motor, which has been waiting for this moment, sees the open track, it latches on, and the engine fires.
This leads us perfectly into the engine room itself. Segment 2. The molecular mechanism of contraction. This is the famous cross-bridge cycle. I want to walk through this step by step because the actual sequence of events is really specific and kind of non-intuitive.
It is a cycle, a repeating loop. And we usually start the description at what we call the resting state. Now, you might think resting means doing nothing, but biochemically, the myosin head is anything but resting. It's actually cocked.
Cocked like a gun or a set mousetrap.
A set mousetrap is the perfect analogy. In this resting state, the myosin head has already used an ATP molecule. It has hydrolyzed it, split it into ADP and an inorganic phosphate, or pi.
Okay.
But, and this is the absolute key, it hasn't released them yet. It's holding onto that ADP and that phosphate very tightly. The energy released from splitting the ATP is now stored in the protein itself, like a compressed spring.
So the energy investment, the spending of ATP, happens before the movement even begins.
Exactly. The system is preloaded with energy. It's ready to go instantly.
Okay, so that's step 1, the cocked state. Then step 2, attachment. The calcium comes in, the tropomyosin moves, and the myosin head, which has a very high affinity for actin in this cocked state, snaps onto the actin filament.
Correct. The cross bridge is now formed. We are locked and loaded.
Now for the main event. Step 3, the power stroke.
This is where the stored chemical energy becomes physical mechanical force. The first thing that happens is the myosin head releases the inorganic phosphate, the pi.
Just lets it go.
That tiny molecular release is the trigger. It causes a massive conformational change in the protein structure. The neck region of the myosin head, the lever arm, cranks forward powerfully.
And it drags the actin filament with it. How far does it actually move it?
It pulls the actin filament about 10 nanometers toward the center of the sarcomere. Now, 10 nanometers sounds impossibly small.
It does.
But you have to remember this is happening in billions of myosin heads at once, and they're cycling hundreds of times per second. It all adds up to a bicep curl.
And at the end of that 10 nanometer pull, it also drops the ADP.
Correct. So now we've reached step 4, rigidity. This is also called the rigor state. At this point, the myosin head is empty. There's no ATP, no ADP, no phosphate bound to it. And in this state, it is physically fused to the actin filament. It is stuck.
And this is the part that always trips people up. In order to relax, to let go of the actin, you need to spend more energy.
Yes. This is step 5, detachment. A brand new molecule of ATP has to come in from the cell and bind to that empty pocket on the myosin head.
And that binding is what breaks the connection.
The simple act of ATP binding to myosin dramatically lowers myosin's affinity for actin. It causes it to let go.
So ATP isn't directly used to pull. ATP is used to reset the trap.
It is used to let go and then to recock the head. So once the ATP binds and the head detaches, we have step 6, the reset. The myosin hydrolyzes that new ATP back to ADP and pi, and that energy is used to crank the lever arm back to the original cocked starting position. And now it's ready for the next cycle as long as calcium is still present.
This whole cycle explains rigor mortis so perfectly. It's not just some vague stiffening. It's a precise mechanical failure.
It is the most macabre and most perfect proof of the entire mechanism. When a person dies, cellular respiration stops. Your mitochondria stop making new ATP.
So the fuel supply is cut off.
The fuel supply is cut off. At the same time, your cellular membranes start to lose their integrity and leak. Calcium, which was stored in the sarcoplasmic reticulum, starts leaking out into the sarcoplasm.
So the on switch gets flipped.
The on switch gets flipped, exactly. The leaking calcium binds to troponin, which moves tropomyosin. The myosin heads, which were sitting there in the cocked position, they see the open actin sites, they bind, and they perform one final power stroke. They pull.
But
now, there is no new ATP arriving to bind to the heads and make them let go.
So they can't detach.
They cannot detach. Every single cross bridge in the body is locked in that rigid step 4 rigor state. The entire body becomes stiff because the muscles are literally fused at the molecular level, and they stay that way until the proteins themselves start to decompose hours or days later.
It's incredibly grim, but it makes perfect mechanical sense. The engine seizes because the lubricant, the ATP, ran dry.
And speaking of engines, the source text makes this really fascinating comparison when it comes to thermodynamics. We have this idea that biology is somehow perfect or 100% efficient. But is it?
The comparison's to a car engine. A standard internal combustion engine, it says, is only about 20, maybe 25% efficient. So for every gallon of gas you put in, only a quarter of that energy actually moves the car forward. The rest is just wasted as heat.
A lot of waste heat. Muscle, by comparison, is an absolute engineering marvel. It is approximately 50% efficient at converting the chemical energy of ATP into mechanical work.
That's incredibly high performance.
Yeah.
Double that of a car engine.
It's fantastic. But as you notice, we still lose 50% of the energy as heat.
Right.
But biology is clever. It doesn't see that as waste. It uses that heat. That's why you get hot when you exercise. It's the byproduct of all those power strokes, and it's the entire principle behind shivering. When you get cold, your brain sends signals to your muscles to contract rapidly and inefficiently. They're not trying to produce useful movement. They're basically just revving the engine in neutral for the specific purpose of generating that waste heat to warm up your core body temperature. It's a feature, not a bug.
That's brilliant. Okay, so we've built the machine with the sarcomere, and we've explained the engine cycle with the crossbridge. Now we need to understand the driver. We know calcium is the key, but how does the body control the calcium so precisely? This brings us to segment 3, regulation.
This is the wiring harness of the system. We need to get an electrical signal from your brain, down a nerve, and deep into the core of that giant muscle fiber to release the calcium exactly when it's needed.
And the central player here is a structure called the sarcoplasmic reticulum, or SR.
The SR is basically a calcium storage tank. It's this intricate, lacy sleeve of membrane that wraps around every single myofibril.
And its whole job is to store calcium.
Its whole job is to store calcium at incredibly high concentrations. In the resting muscle, it's constantly running these little pumps, the Circa pumps, which use ATP to vacuum calcium out of the cell fluid and hoard it inside the SR tank. This keeps the sarcoplasmic calcium levels extremely low so the muscle stays relaxed.
So how does the signal from the nerve get to the tank to tell it to release the calcium?
The text says the mechanism is different depending on the muscle type.
Very different, yes.
In skeletal muscle, it describes something called mechanical coupling. This sounds almost Rube Goldberg-ish, like a physical lever.
It effectively is a physical lever. It's amazing. So the nerve signal, the action potential, travels along the muscle cell's outer membrane, the sarcolemma, but then it dives deep into the cell through these little tunnels called T-tubules.
T for transverse. They run across the fiber.
Right. And embedded in the wall of this T-tubule is a special protein, a voltage sensor, called the dihydropyridine receptor.
Okay.
When the electrical signal comes zipping down the T-tubule, this sensor protein physically changes its shape. And right across a tiny gap from it, embedded in the wall of the SR calcium tank, is the release valve. That's the ryanodine receptor, or RYR1.
And here's the mechanical part. In skeletal muscle, the dihydropyridine sensor is physically tethered to the ryanodine valve. They are bolted together. So when the voltage sensor moves, it literally pulls the plug out of the SR tank.
That's incredible. It's a direct mechanical linkage. That explains why skeletal muscle can react so incredibly fast.
It's almost instantaneous. But cardiac muscle, your heart, does it differently. The text is very clear on this distinction.
The heart uses what it calls chemical coupling, or calcium-induced calcium release, CICR.
Right. In the heart muscle, the T-tubules and the SR are not physically bolted together. There's a small gap. So when the action potential comes down the T-tubule in the heart, the dihydropyridine receptor acts as a true calcium channel. It opens up and lets a tiny little bit of calcium enter the cell from the outside.
So a little splash of calcium comes in from the bloodstream.
Just a little puff, a splash. And that small amount of calcium diffuses across the gap and binds to the ryanodine receptor on the SR tank. That binding is the chemical signal that triggers the main valve to open and dump the main tank of calcium.
So it's like using a match to light a bonfire.
That's the perfect analogy. The match is the small amount of calcium coming from outside, but the fire is the huge release from the SR inside.
Whereas skeletal muscle doesn't need an external match. It just has a flint and steel built in.
Exactly. And this is clinically very important. It means your heart's ability to contract is very sensitive to the calcium levels in your blood. If your blood calcium gets too low, your heart can fail to contract properly. Your biceps, on the other hand, don't really care about the calcium outside the cell because they rely on that direct mechanical pull.
It's such a subtle but critical difference. Let's briefly touch on smooth muscle regulation because it's the real outlier. We said before, no troponin, no striations. So how on earth does it know when to contract?
It uses a completely different pathway. Calcium is still the trigger, but it works on a different target. When calcium enters a smooth muscle cell, instead of binding to a lock on the actin tract, it binds to a free-floating protein in the cytoplasm called calmodulin.
Calmodulin. Okay, so what does the calcium calmodulin team do?
They go hunting for an enzyme. This enzyme is called myosin light chain kinase, or MLCK. The calcium calmodulin complex finds MLCK and activates it.
Okay.
This activated enzyme then goes over to the myosin motor itself and adds a phosphate group to one of those little light chains on its neck.
It phosphorylates the myosin head.
Yes. In smooth muscle, the myosin head is naturally lazy. It's off. It won't grab actin at all unless it has that phosphate stamp of approval on it. So the on-off switch isn't on the track like in skeletal muscle. The switch is on the motor itself.
That seems like a much slower, more deliberate process.
It is much, much slower to initiate a contraction. But it also allows for that latch state we mentioned. Once the muscle is contracted, another enzyme, a phosphatase, can come along and remove that phosphate. But the myosin heads can actually stay stuck to the actin for a long time, maintaining tension with almost zero ATP usage.
Which is perfect for holding your blood pressure steady all day.
Efficiency is the name of the game in smooth muscle.
Which brings us directly to segment 4, metabolism, the fuel. The text drops a really shocking statistic. Muscle only stores enough ready-to-use ATP for a few seconds of contraction.
It's a classic just-in-time inventory strategy. ATP is a big, bulky molecule, and it's chemically unstable. If you try to store enough ATP for a marathon directly in your muscles, your legs would be the size of tree trunks. It's just not efficient.
So instead, you store more stable fuel sources, and you generate the ATP on demand.
As you need it, second by second.
So let's run through the fuel tanks, from fastest to slowest. The first line of defense is creatine phosphate.
The backup battery. This is your immediate reserve. We have a pool of this molecule, creatine phosphate, floating around in the muscle. It holds a high-energy phosphate bond. And we have an enzyme called creatine kinase, or CK.
And what does CK do?
As soon as you burn one molecule of ATP and make ADP, CK grabs the high-energy phosphate from creatine phosphate and slaps it right back onto the ADP. Boom! You've instantly regenerated an ATP.
How long can this system last?
It's near instantaneous, but the tank is very small. It'll last you maybe 10-15 seconds of all-out effort. This is your 100-meter dash fuel. It's your lift a really heavy box off the floor fuel.
Clinically, creatine kinase is important too, right?
Yeah.
Doctors measure it in the blood.
Yes, because CK is normally inside muscle cells. If you have a heart attack, the heart muscle cells die and burst open, releasing their CK into the bloodstream. So a spike in a specific type of CK is a major marker for a heart attack.
Okay, so after 15 seconds, the creatine phosphate tank is dry. What's the next system to kick in?
Glycolysis. Your body starts breaking down glucose sugar that's either in your blood or stored in the muscle as glycogen. This happens right there in the cytoplasm. It's very fast. It's like kicking on a turbocharger, but it's inefficient and it's dirty.
Dirty because it doesn't use oxygen. It's anaerobic.
Exactly. It's anaerobic. And the byproduct is lactate, which gets converted to lactic acid. This buildup of acid in the muscle contributes to that burning sensation and fatigue. Glycolysis can get you through maybe a minute or two of intense effort, like a 400-meter sprint.
And then for the long haul, the marathon runner, the Tour de France cyclist.
For that, you need the main power plant, oxidative phosphorylation. This happens in the mitochondria. You're feeding oxygen, fatty acids, and the breakdown products of glucose into the Krebs cycle and the electron transport chain.
And this is much more efficient.
It's much slower to ramp up. It's like a big diesel engine, but it burns very clean, just producing CO2 and water, and it generates massive amounts of ATP. This is the system that can keep you moving for hours on end.
The text mentions one more, kind of a desperate measure enzyme that I found fascinating, adenylyl kinase.
Yes. The last resort enzyme. I love this mechanism because it's so clever. So imagine the cell is in dire straits. It has burned through all its ATP. It's used up all its creatine phosphate. The cell is just swimming in used up ADP.
Okay.
Adenylyl kinase is an enzyme that takes two of those ADP molecules and smashes them together.
What does it get?
It transfers a phosphate from one ADP to the other, so you get one regenerated ATP molecule, which keeps the cell alive for one more power stroke, and one AMP molecule, adenosine monophosphate, which only has one phosphate.
And that AMP is the real point of the reaction. It's an SOS signal.
It is a screaming red alert for the cell. High levels of AMP tell the cell's metabolic machinery, we are in an energy crisis. We are dying here. Ramp up glycolysis, burn more sugar, start building more mitochondria. It's the molecular trigger that drives fitness adaptation. When you train to failure, you're generating AMP, and that's the signal that tells your body to get stronger and more efficient for next time.
And this whole fueling strategy is reflected in the different fiber types, isn't it? The text makes a clear distinction between type I and type 2 fibers.
It does. They're specialized for different metabolic strategies. Type I, or slow-twitch fibers, are your red fibers. Think of the dark meat on a chicken or a turkey leg.
Why are they red?
They're red because they are packed with a protein called myoglobin, which stores oxygen, and they're full of mitochondria. They have a rich blood supply. They are built for endurance, for oxidative metabolism. They don't generate huge force, but they are very resistant to fatigue.
These are your posture muscles, the ones that keep you upright all day, or a marathoner's leg muscles.
Exactly. Then you have type 2, or fast-twitch fibers. These are your white fibers, the breast meat of a chicken.
Which can't fly very far.
Exactly. A chicken can flap its wings furiously for a few seconds, but that's it. These fibers have less blood flow, less myoglobin, and fewer mitochondria, but they are packed with glycolytic enzymes and have huge stores of glycogen and creatine phosphate.
So they're built for power.
Massive, explosive power. But they burn out in seconds. These are a sprinter's muscles, or a weightlifter's.
It's amazing how specialized this system is. Okay, so we've built the machine, we've wired it up, and we've fueled it. Now let's talk about what happens when it breaks. Segment 5. The clinical correlates. The text highlights some truly terrifying conditions where these specific molecules fail.
And probably the most dramatic and immediate is a condition called malignant hyperthermia. This is an operating room nightmare scenario.
It's triggered by certain kinds of anesthesia, right? The text mentioned halothane, or a muscle relaxant called succinylcholine.
Yes. In people who have a specific genetic mutation in that ryanodine receptor, the RYR1, remember that's the calcium floodgate on the SR tank.
I remember.
For these susceptible people, those anesthetic drugs act like a key that gets stuck in the lock and jams it wide open.
The calcium tank just empties into the muscle uncontrollably.
The floodgates burst open. Calcium pours out into the sarcoplasm all over the body. The muscles contract and become rigid. The circuit pumps go into overdrive, trying to vacuum all that calcium back in, which burns through ATP at a catastrophic, unbelievable rate.
And burning that much ATP that fast must generate an incredible amount of heat.
Massive amounts of heat. That's the hyperthermia part. The patient's body temperature skyrockets. They become rigid as a board. They start producing huge amounts of acid from anaerobic metabolism and CO2. They are literally cooking from the inside out.
It's a complete runaway engine.
A total metabolic meltdown. And the cure is fascinating. It's a drug called dantrolene. It's one of the few specific antidotes we have in medicine. It works by binding directly to the ryanodine receptor and physically plugging the leak. It stops the calcium release at its source.
Wow. Next on the list, muscular dystrophy, specifically Duchenne MD. You said this isn't an engine failure, it's a chassis failure.
It is a profound structural failure, and it involves a protein we haven't mentioned yet, a protein called dystrophin.
Which is a monster of a protein, right?
The dystrophin gene is the single largest gene in the entire human genome. It's enormous. And its job is to be a molecular shock absorber and anchor. It connects the actin cytoskeleton inside the muscle cell to a complex of proteins in the cell membrane, which then connects to the connective tissue outside the cell.
So it physically links the contractile engine to the outside world and reinforces the membrane.
It does. It protects the delicate cell membrane from the sheer mechanical violence of contraction and relaxation. In Duchenne muscular dystrophy, patients have a mutation that means they make no functional dystrophin protein at all.
So every time they move, every time it also contracts.
Every single time the muscle contracts, the membrane, lacking that reinforcement, tears. It develops little micro tears all over it.
And if the membrane tears, it's no longer a perfect barrier.
And things leak. Specifically, calcium from the outside world, where it's in high concentration, floods into the cell. And as we've established, uncontrolled calcium inside a cell is toxic. It activates enzymes that chew up the cell's proteins. The muscle fibers die, one by one, and are slowly replaced by fat and scar tissue.
Which explains the progressive weakness and also that characteristic look of large calves, the pseudohypertrophy. Looks like big muscle, but it's actually scar tissue.
It's scar tissue and fat. It's heartbreaking because the engine, the actin and myosin, works perfectly fine. The fuel systems are fine. But the casing is defective, and it just tears itself apart with use.
And Becker MD is the milder version of that.
Yes. In Becker muscular dystrophy, the mutation results in a dystrophin protein that is present, but it's altered. Maybe it's a bit shorter or misshapen. It works a little bit, so the tearing is slower and the disease onset is much later in life.
The third clinical link the text draws is with cardiomyopathies, or diseases of the heart muscle.
Right. Specifically, a condition called familial hypertrophic cardiomyopathy. This is often caused by one of several hundred different mutations in the beta-myosin heavy chain gene.
So a defect in the motor itself.
A defect in the cardiac engine. The heart muscle tries to compensate for this inefficient motor by getting thicker and thicker. That's the hypertrophy. But the new muscle is laid down in a disorganized, chaotic way.
And that's dangerous.
It's extremely dangerous. It's the leading cause of sudden cardiac death in young athletes. You hear about a high school or college basketball player who just drops dead on the court during a game. Very often, this is the underlying cause. The heart muscle is big and strong, but the electrical signals that coordinate the heartbeat get lost in that disorganized scar tissue, leading to a fatal arrhythmia.
It is so sobering to realize how fragile this whole system is. A single amino acid change in one of the biggest proteins in your body can stop your heart.
It's both incredibly robust and exquisitely delicate at the same time.
Let's pivot to a slightly happier clinical story, or at least a more famous one. Nitric oxide, segment 6. This whole story reads like a medical detective novel.
It really does. For decades, physiologists knew this weird fact. If you took a blood vessel out of an animal and put a chemical like acetylcholine on it, the smooth muscle in the vessel wall would relax, causing vasodilation.
Okay.
But it only worked if the inner lining of the blood vessel, the endothelium, was intact. If you were clumsy and scraped off that single layer of endothelial cells, the acetylcholine, nothing. The muscle wouldn't relax.
So the lining was secreting some kind of mystery messenger that was telling the muscle what to do.
Exactly. And for years, they just called it EDRF, endothelium-derived relaxing factor.
Which is just the most uninspired scientific name ever. The thing that does the thing.
Scientists can be very literal sometimes. But eventually, in a discovery that won the Nobel Prize, they figured out that EDRF was something incredibly simple. It was nitric oxide. NO, a simple gas.
How does a simple gas control a complex muscle?
It's the elegance of physics. When the endothelial cells are stimulated, they make NO from the amino acid arginine, using an enzyme called NO synthase. Now, because NO is a tiny, uncharged gas molecule, it doesn't need channels or receptors to cross membranes.
It just diffuses.
It goes right through the cell walls, from the endothelium into the neighboring smooth muscle cells. It's an instantaneous signal.
And once it's inside the muscle cell, what does it do?
It finds and activates an enzyme called guanylyl cyclase. This enzyme's job is to take GTP and convert it into a messenger molecule called cyclic GMP or cGMP.
And cGMP is the ultimate relax command for the muscle.
Yes. High levels of cGMT activate a cascade of other enzymes that ultimately lower the intracellular calcium levels and activate the phosphatase that removes the phosphate from myosin. The muscle relaxes, the blood vessel widens, and blood flow improves.
This is the exact mechanism of nitroglycerin, right? The little pills people put under their tongue for angina.
It's exactly that. Nitroglycerin is a prodrug. Your body chemically breaks it down, and it releases NO. The NO relaxes the coronary arteries, allowing more oxygen-rich blood to reach the starving heart muscle and relieve the chest pain.
Well, we have to mention the other blockbuster drug that works on this very same pathway.
We do. Sildenafil, better known by its trade name, Viagra.
The text explains that it works on the cGMP pathway, but at a slightly different point.
Right. Viagra doesn't help you make more NO or more cGMP. It works on the other end of the signal. There's another enzyme in the cell called phosphodiesterase 5, or PDE5. Its only job is to find and destroy cGMP to turn off the relaxation signal.
So sildenafil inhibits that enzyme. It stops the stop signal.
Precisely. It prevents the breakdown of cGMP, so when a relaxed signal does come along, the cGMP hangs around for much longer, the signal's amplified, and you get more sustained vasodilation and increased blood flow. It was, as many know, originally designed as a heart medication for angina.
But patients in the clinical trials started reporting specific unexpected side effects.
And a billion-dollar industry was born. It's the ultimate proof that one single molecular pathway can have very different physiological applications depending on where in the body you target it.
It's all about context.
Amazing.
We have a little bit of time left, and we absolutely need to cover the structure that holds all of this together and so much more. Segment 7, the cytoskeleton. We've focused so heavily on muscle, but the text makes a point to say that this machinery is in every single cell.
The cytoskeleton is truly the unsung hero of cell biology. And it isn't just a passive scaffold like the studs in a wall. It's a dynamic, living structure. It has three main components: microfilaments, microtubules, and intermediate filaments.
We've already met microfilaments. That's just another name for actin.
That's right. In non-muscle cells, actin forms networks and stress fibers that give the cell its shape and allow it to crawl around. But it's also absolutely crucial for cell division. When one cell needs to split into two, a process called cytokinesis, it forms a ring of actin and myosin right around its middle.
And that ring contracts.
It contracts like a purse string or a drawstring on a bag. It cinches the cell membrane tighter and tighter until it pinches the one cell into two separate daughter cells. Without actin, complex life as we know it couldn't exist because our cells couldn't divide.
Let's move on to the second type, microtubules. The text calls these the highways of the cell.
That's a great description. Imagine long, hollow tubes made of a protein called tubulin, and they are incredibly dynamic. They are constantly growing and shrinking by adding or losing tubulin subunits at their ends. We call this dynamic instability.
And they act as a rail network for transport inside the cell.
Yes, a cell is an incredibly crowded place. You can't just wait for important molecules or organelles to randomly float to where they need to go. You need an active transport system.
So we have molecular trucks?
We have molecular trucks. Motor proteins called kinesin and dynein physically walk along these microtubule tracks, carrying cargo vesicles from one part of the cell to another.
When you say walk,
yeah,
you mean that metaphorically, right?
I mean it literally. If you look at the high-resolution animations based on crystallography data, these proteins have 2 feet, which are globular heads that bind to the tubulin. They step over each other, 1 foot in front of the other, burning a molecule of ATP with each step as they drag their cargo along. It's both adorable and completely mind-blowing.
And kinesin and dynein walk in different directions.
Yes. The microtubule tracks have a polarity, a plus end, and a minus end. Kinesin generally walks toward the plus end, out towards the edge of the cell. Dynein walks toward the minus end, in towards the center.
And these same microtubules also form the core of cilia and flagella.
They do. The long tail of a sperm cell, the flagellum, is a bundle of microtubules arranged in a very specific 9+2 pattern. And they move because dynein arms, which are attached to one microtubule, walk along the neighboring microtubule, causing the whole structure to bend and create a whipping motion.
The text mentions a genetic condition here, Carter-Giner syndrome.
Yes, this is a form of primary ciliary dyskinesia. If you have a mutation that creates defective dynein arms, the microtubules can't slide past each other. Your cilia and flagella can't move.
So what does that mean for the patient?
Well, you have cilia all through your respiratory tract, and their job is to beat constantly to sweep mucus and debris out of your lungs. If they can't beat, that mucus builds up. So these patients suffer from chronic severe lung infections.
Okay.
And in males, the flagellum on the sperm can't swim. So they are infertile. It's a classic example of how one single molecular flaw in one motor protein can connect to seemingly completely unrelated symptoms, chronic lung disease and male infertility.
And microtubules are also a major target for some heavy-duty cancer drugs.
Because they are absolutely essential for cell division. During mitosis, microtubules form the spindle that pulls the duplicated chromosomes apart into the two new daughter cells. If you mess with the tracks, the cell can't divide.
And drugs like Taxol and vinblastine do exactly that.
They do, but in opposite ways. Vinblastine and another drug called colchicine work, but preventing microtubules from assembling, they dissolve the tracks. Taxol, or paclitaxel, does the exact opposite. It stabilizes the microtubules so they become too rigid and can't break down. It freezes the tracks in place.
But the end result is the same.
The end result is the same. The cell tries to divide, it sees that it can't move its chromosomes properly, and it triggers a self-destruct program called apoptosis. That's how we target and kill rapidly dividing cancer cells.
Okay, finally, the third component. The intermediate filaments. These are the structural beams, the rebar.
They are. They provide tensile strength. They aren't dynamic like actin or microtubules. Their job is to be strong and durable and resist stretching. And what's interesting is that different cell types make different kinds of intermediate filaments.
Which is useful for doctors.
Very useful for pathologists. If they find a tumor, they can test to see what kind of intermediate filament it's making. If it's making keratin, they know the tumor originated from epithelial tissue like skin or lining cells. It's a carcinoma. If it's making desmin, it came from musculosarcoma.
And mutations in keratin itself cause disease, right?
Yes, a group of devastating skin diseases called epidermolysis bullosa. The keratin filaments in the skin cells are defective, so the cells can't hold on to each other or to the underlying tissue. The slightest touch, even just friction from clothing, causes the layers of the skin to shear apart and form horrible blisters. It's a disease of pure structural failure.
And the final example, inside the very nucleus of the cell, we have filaments called lamins.
Which brings us to our final and perhaps most profound clinical point, progeria, or Hutchinson-Gilford progeria syndrome.
This is the disease that causes rapid premature aging in children.
Yes. These children often look like little elderly people by the time they are 10 or 12 years old. And it's caused by a single point mutation in the gene for lamin A, which is the intermediate filament that forms a mesh-like lining on the inner surface of the nuclear membrane.
So a structural defect in the lining of the nucleus causes the whole body to age. How does that work?
The thinking is that the physical integrity of this nuclear lamina is vital for organizing the chromosomes, for regulating gene expression, and for repairing DNA damage. When the nucleus is structurally floppy or unstable because of the bad lamin, the genome itself starts to degrade, and the whole organism undergoes what looks like an accelerated aging process. It's this incredible link between mechanics and genetics.
We've covered a truly massive amount of ground today. We've gone from the sliding of filaments in your bicep all the way to a gas that regulates your blood pressure to the literal highways and support beams inside every one of your cells.
It really highlights the so what of biochemistry, doesn't it? We aren't just memorizing the names of proteins for an exam. We are learning the actual instruction manual for the machine that you live in every single day.
It connects the micro to the macro in a way I hadn't really appreciated before. A single protein failing and your whole body fails.
And that's the thought I want to leave the listener with. We talked about how incredibly robust this system is, 50% efficiency, cycling billions of times over a lifetime. But we also saw how exquisitely fragile it is. A single amino acid change in a dystrophin gene, or a myosin gene, or a lamin gene, and the entire system can collapse.
Evolution has built a Ferrari, but the engineering tolerances are incredibly tight.
Incredibly tight. So the next time you go for a run or you lift something heavy or you even just pick up your coffee cup, try to visualize it.
Visualize that flood of calcium.
Visualize those billions of myosin heads snapping onto the actin, ratcheting back and forth, burning ATP, releasing phosphate, pulling that filament just 10 nanometers at a time. It is a silent, invisible, and absolutely magnificent symphony happening under your skin right now.
And with that incredible image, we'll wrap up. Thanks for listening to The Deep Dive. Keep those engines running.
Stay curious.